Oh, really?
I don't want that to attack you. It seems like it's gonna. There we go. Solid. I've now finished all of the introductions that have come in, and I am very honored to be working with a really rad group of people. Thank you for sending those my way, and I hope that I can integrate your passions and interests in every way into microbiology. I know that some of you have already expanded your horizons into the YO courses shell, and you've found all of the resources on there. How many of you have already found homework number 1? It's a PDF file. Nice! You guys rock. Bring that to class, and ask me questions from the homework while we're covering the material, because it's a sweet way to get your homework done. We will talk explicitly about every question on the homework. Now, you may think, gosh, Rachel, I don't remember covering this, but I promise that we did. It's that I'm asking you to apply and think about the concept that we've covered. So if you find yourself feeling a little confused on a homework question, don't hesitate to ask me. Don't hesitate to ask in class or to come to office hours and get that homework finished off in a way that you feel good about before entering your answers. Once you feel like, yeah, I've done a great job on this homework, I'm ready to go in and enter your answers onto an answer sheet within the YO Courses shell where you found the PDF file. Okay, if you accidentally click on that before you're ready to enter your answers, just let me know and I will reset it for you. But please don't go in there and try to do it before. Did you do it already? No? Oh, good. Okay.
So I'm trying to just get a schedule thing on.
Yep.
So we have Learn Smarts that are due like.
Okay, cool. So you have nothing due on any day but Tuesday. You got it. Only Tuesday, ever Tuesday. Everything else is extra credit, right? So Learn Smart is totally extra credit. The way we jive with that is that I release chapters as we kind of cover them, and it's sort of random how I set the due date. And typically with extra credit, you'll let me know if you're keeping up. We'll reset the due dates within reason. If we get to a point that we have passed the material a long time ago, it seems not relevant anymore to reset those due dates. So please do remember that the only required thing is the homework that's due on Tuesday, and of course the exams that we take on the Tuesdays that we don't have homework. Learn Smart is not required. It's an extra credit activity. If you want to jazz with that, go in, get that, and submit them at the dates that I said I've set them. You know, unless for some reason they don't seem reasonable, let me know, I'll reset them, okay? So does that help? Yeah, and I think it's really important to just differentiate the kind of clutter in your mind right now between what's extra credit, like what's that 30-point extra credit, versus what's really required. So find that first required homework, definitely want to get that done, and certainly stop me with questions. Also, if you haven't had a chance to do so, watch that podcast on the current events poster. Most of you did, because you incorporated it into your introductions. I loved it. It was fantastic. So labs will begin tomorrow. Now, here's the good news. You don't really have to have anything for tomorrow. However, if you want to bring your lab book, if you did purchase it, it will be helpful for you to follow along with the kind of safety and etiquette that we'll cover throughout that process. So definitely not a big stressful lab tomorrow, but beginning the next week, we'll start to have pre-labs and everything will get a little bit more real, okay? Any other questions before we jump into microbiology? We actually get to do that today. Okay, cool, because that's where I want to go. So today we are going to be talking about the history of microbiology, the implications and the applications, and I'm going to integrate in some of the questions that you all have come up with on the Follow Your Gut thread. So I want you to ponder this question. How many years ago did scientists accept or fully support that all life arises from other living things of the same kind? Now, this sounds pretty obvious to us. In kindergarten, at some point, you learned that, you know, older flies have baby flies and they come from the older flies, right? You knew that somehow life begets life. But that wasn't always known. And in fact, for a long time, people thought you could have just completely inert inorganic material and that from that life could arise. They called it spontaneous generation. And people really adhered to that for a long time. So I want you to take 1 minute, roughly 1 minute, to talk to the person next to you and come up with an answer to this. Write it down somewhere, okay? Chat with somebody by you. I love this. I'm hearing so many wonderful things like, wasn't that the Dark Ages? Write down your answer and keep it tucked away in your mind. And we are going to come back to this, okay? So make sure you have something, maybe you've argued with your neighbor and you kind of have convinced him or her that you're right, and you've written down an answer. Tuck that away. Well, I want to go ahead and look back quite a ways, in fact, to the beginnings of the Earth. Some 4.5 billion years ago, the Earth is thought to have come into existence. And of course, life on Earth, about 3.5 billion years ago, is where we have fossil record. In 1977, there was a fossil of an Archean chert found in Australia, and it was dated back 3.5 billion years. So life has been around for a very long time. Microorganisms have existed for some 3.5 to 3.8 billion years. Interestingly, it took us a very long time to realize that. And it wasn't until a man named Antonie van Leeuwenhoek, who was from the Netherlands. He was a haberdasher and a draper by day, but by night he was a superhero who built microscopic lenses, and he made lenses that were capable of magnifying much more than the lenses of Hooke or Galileo. They can magnify up to 300 times, and that meant that for the first time, Antonie van Leeuwenhoek was able to see a world that no one else had ever seen. Imagine what that must have been like, to see all of these itsy-bitsy little tiny animals. He called them animalcules. And he wrote about them with wild abandon. He was extremely excited. I mean, you guys know how this goes. Who's seen an amoeba on a microscope before? This is rad stuff. But imagine being the first person to really be describing it. He got so jazzed that he went around everywhere and he collected samples. He would collect samples of well water. He would collect samples of urine, and he would look to see what he could see swimming around in them. And then he would write these incredible descriptive letters to the Royal Society of London. He wrote, for example, very many little animalcules, whereof some were roundish, while others a bit bigger consisted of an oval. On these last I saw 2 little legs near the head and 2 little fins at the hindmost end of the body. Others were somewhat longer than an oval, and these were very slow a moving and few in number. These animalcules had diverse colors, some being whitish and transparent, others being green with very glittering little scales. Others again were green in the middle and before and behind white. Others yet were ashen gray. And the motion of these animalcules in the water was so swift and so various, upwards, downwards, and roundabout, that it was wonderful to see, right? How jazzed was he? I mean, this is good stuff. So the Royal Society of London began to compile his works and letters, and we might say, although others had seen some microscopic life, that Antonie van Leeuwenhoek was really the father of microbiology. And in many ways, he is attributed that title. And maybe we can say that the birth of the study of organisms that were so small began. This is an amoeba. Maybe some of you have seen that before. This is a Ciliaphoria stentor. You can see its cilia moving like crazy. We'll see some of these things under the microscope tomorrow, and that was what Antonie van Leeuwenhoek was seeing. So the study of life forms and agents that are too small to be seen with the unaided eye. Now, clearly, we know we study other things in microbiology that don't quite fit that definition. For example, we like to dork out to the fungus among us, and many of them are very macroscopic. We see mushrooms with the naked eye, and yet they fit into the category of microbiology. So inevitably, there are things that will fall outside of this definition. Always is the case, but it is kind of how we think about microbiology. Now, it's interesting to tell you, if you were in my class 10 years ago when I was giving this first day's lecture, this definition would have been different. I would have told you that it was the study of life forms, no agents there. Does anybody know what the agents describes? Yeah, Riley. Yeah, viruses, good. So retroviruses are one type of virus. That's perfect, yeah. So viruses, what else would be in there? Agents. Thoughts on that? Well, bacteria are actually considered life forms. But here's the rub. This is interesting. We need to think about whether we want to get into the argument of where the line of life is. Because last semester there was actually a really cool article published arguing that perhaps we need to rethink certain viruses as being life forms. So that's, you know, an interesting question of where that line occurs. What else fits in there? Yeah, good. Prions or prions, depending upon your pronunciation, are simple proteinaceous infectious agents. Who's heard of CWD? Chronic wasting disease, or BSE, bovine spongiform encephalopathy, also known as mad cow, right? Those are also agents. So there are other things that fit into this category of microbiology. And I just want to make a quick note of what are these life forms versus what are these agents. And I promise that we will be talking more about each one. So in our top picture, this is actually something that you're very soon going to be seeing in lab. And the cells right here are going to interest a number of you. I know Sean, and I was saying your name wrong last time. Are you in a different place today? Of course you moved, right? Likes to brew beer, right? And I'm going to pick on Crystal a little later and interested in wine. So these are yeast. This is actually Saccharomyces cerevisiae. And yeast, of course, are a eukaryotic life form. So yeast would fit into that category of life form. Saccharomyces cerevisiae. Can anybody see right here these itsy bitsy little tiny guys? They're little bacteria. And bacteria are, of course, life forms. And this particular bacterium is an extremely famous one, Escherichia coli, or E. coli. You hear a lot about that one. Over here on the right, this is actually Volvox, which is a protist. It's a type of algae for the common name. So protists are considered life form. Protists actually encompass a wide range of different things, not just algae. But we can also add to this fungi, of course, are life forms. And besides eukaryotic organisms like fungi and protists, like yeast, and then, of course, bacteria. Can anybody think of another life form out there? Yeah. Archaea, you got it. Sweet. Why am I forgetting your name today? Help me. Jake. So Jake is on today, and archaea are another form of life. These are what we might consider the ancient ones or the extremophiles. Not all of them are extreme, but by our estimation, many of them like to live in pretty crazy places, like in Antarctica, in the really, really cold, or in the really, really hot, or in really acidic environments. So these are our life forms. And then, as you all told me, agents include things like viruses. This is an epically famous virus. Oh, and who was it? Help me, said, oh, name, you are interested in HIV. Was that Drew? Was that you? Yeah. Okay, is that the name right? Yeah, oh, sweet. So absolutely, HIV, that's what we've got there. And then our prion, proteinaceous infectious agents like the chronic wasting disease protein or the BSE protein. And we hear less about these, but these are viroids. They're RNA and can cause disease. There's also something called satellites that, when accompanied by a virus, is just a RNA or DNA, but together with virus, they cause disease. So it's amazing how many things there are out there that can be infectious agents or that can be life forms, right? Not everything is infectious. Some things are obviously good. So many, many things. So this is a wide swath of what we see in the definition of microbiology. So let's talk a little bit more historically, however, and we'll take a moment to return to this journey of fighting the notion of spontaneous generation. So spontaneous generation would quite literally say that if I had this little thing of broth right here, and spontaneous generation were true, if I just set this on the shelf, now do you know that we have autoclaved this broth, meaning that we've raised it to high temperature and pressure so that we've killed everything in there? Spontaneous generation would hold that the life could still pop up in here spontaneously, like from nothing. Well, I guess it's not nothing, right? It's broth. That life could just pop up in there. But in fact, many scientists wanted to fight this notion of spontaneous generation, and they wanted to try to show that in fact life comes from other life. That is biogenesis. All life arises from other things that are living of the same kind. And one of these men is the man who is pictured right here, Francisco Redi. This is a photograph that I took when we were in Italy for the World University Games. We took the ski team to Florence, and it turns out that Francisco Redi was one of the most auspicious Florentinians. He was incredibly famous scientist of Florence. And what he studied was flies. He was super jazzed on flies. And he would actually watch all of their behaviors. And one of the things that he wanted to see is whether or not flies came from maggots. And he wanted to see if he could take a piece of meat. Now, forgive me, this is not actually a slab of beef, but instead I just used a piece of beef jerky. So assume this is a slab of beef, right? And he put it in a jar, and he had 3 different jars. He had one that was completely uncovered, like this. So what would probably happen to this meat if you left it uncovered on the shelf? It would rot, right? Flies would lay their eggs, or it'd be maggots, it would putrefy. Gross. Blech, okay? But what if Francisco Redi took a piece of gauze, and this is actually what he did, and he put this gauze over the top of the jar? Now, what couldn't happen? The flies, right? And so where would the flies lay their eggs? Well, they actually, it's interesting, they actually do. They lay their eggs right on top of the gauze. So you could see all the eggs are up there and not on the meat. So you're starting to separate out, you know, the meat just causing the growth or the putrefaction versus the flies, right? So this is the idea that we're trying to get at, disproving spontaneous generation, because it's the eggs of the flies, right, that cause the putrefaction and not the meat itself, right? It's not just that the meat spontaneously grows stuff. And so really, in a lot of ways, now what if he just covered it with a top like this? Then the flies couldn't even smell it, right? And in fact, I should put that down, right? You know, you completely tightly, and they wouldn't lay their eggs there now, yeah. So it's interesting that in the late 17th century, in fact, Francisco Redi's work should have caused us to say, hey, baby flies come from adult flies. Life comes from other life. And it's not the meat that's causing the random popping up of growth, but in fact, it's the eggs of the flies that are being laid on that meat. Life is coming from other life. But unfortunately, what couldn't Francisco Redi prevent with his study? Yeah. And so this led many scientists to continue to hold to the idea of spontaneous generation. They were like, there still must be something spontaneous going on the meat, allowing the meat to go bad, right? It must be generating life spontaneously. And so think of this, years and years and decades passed. And many scientists still tried to fight this notion of spontaneous generation. They would do things like boiling their nutrient broth, sealing it in a flask, and showing that nothing would grow. But still, people said, well, okay, yeah, sure. What happens is when you boil that broth, you take some vital force out of the air, and you stop its ability from being able to allow spontaneous generation. Right? And so there are all these arguments trying to hold on to spontaneous generation. Somebody named Felix Pouchot even showed that, in fact, you could take air completely away and there would still be growth. So it must not be the air that was allowing spontaneous generation, yet people still held to spontaneous generation. So this is crazy. And in fact, I want to read you a quote from an article written by John Tyndall. And he speaks in 1871 about the way in which the world was divided. Okay, so check this. You know that as regards the lowest forms of life, the world is divided and has for a long time been divided into two parties. The one affirming that you have only to submit absolutely dead matter to certain physical conditions to evolve from it living things. The other, without wishing to set bounds to the power of matter, I love that part, affirming that in our day no life has ever been found to arise independently of pre-existing life. Many of you are aware that I belong to the party which claims life as a derivative of life. Any thoughts on that passage? This is one of my favorites, because it makes me think about like where we are now and how we describe, oh, some people sit in this party that believes this, and other people sit in this party that believes that, and we're divided and we're really dual about our opinions on this. At one point, that's how we felt about whether life came from other life. Like that's how incredibly binary or polarized this issue was. So onto the scene in the late 19th century comes a very famous man whom you might recognize as being a famous scientist in many ways, and this was Louis Pasteur. So Louis Pasteur comes about and he says, okay, come on guys, really? You know, really, are we still thinking that things spontaneously generate? How could we show that air can get to this sterile broth, but that air doesn't just cause spontaneous generation? So he ingeniously designed a flask. This flask had a swan neck on it. So looking at the design of this flask, what was his genius? What happened when you had sterile boiled broth in here and you expose it to air through the swan neck? What happened in that first U-bend? Yeah, all the bacteria and all of the particulate matter in the air dropped out. So that first U-bend caught all of the microbial life. But what didn't it stop? Air. So air still got through, but bacteria and other microorganisms did not. And so when Louis Pasteur left this sterile broth on the shelf for years, it stayed sterile. Does that make sense? So he was able to show that spontaneous generation in the presence of air would not occur because he was separating out the microbial world from the air. So Pasteur's contributions here were incredibly valuable because he was showing in one fell swoop that microorganisms were found in the air, but that these could be separated from the air itself, and that the air itself didn't cause spontaneous generation. However, if he broke the top of this swan-necked flask, then all of a sudden, the microorganisms could fall right into the broth, and within a day, growth would occur. So the two major contributions that we could say that Louis Pasteur made with this experiment were, one, microorganisms are in air. I'm running out of room. This is terrible. Microorganisms are in air. And in fact, he did this a couple of ways. He passed air through a cotton plug, and then he looked at that cotton plug under the microscope, and he was able to show, yeah, there's microorganisms in there. And then he could say, okay, well, I'm going to separate those out with this first bend of the flask. And then two, this is the big one. There is no elusive vital force in air to cause spontaneous generation. Which is kind of what it's what the spontaneous generation fraction of the world thought would be found in air, right? This vital force in air that allowed spontaneous generation. Because he could allow exposure to that air without seeing any growth. So here we are at this beautiful place where everyone in the world should have said, woohoo, yeah, Louis Pasteur, we all now believe in biogenesis. But science is fickle. And what are the couple of things that we know have to be true of any scientific experiment to make it good science? Say it one more time, Sarah. You have to do it a lot. And dang nubbit, this is where everything fell apart. Because all these other scientists were like, oh, okay, all right, okay, you know, you did it, Louis Pasteur, good job. I'm just going to replicate your work for good measure. And it turns out that people couldn't replicate his work. And so they would do this experiment again, and they couldn't, and they would get growth. And they would say, well, I don't know how you ended up with a sterile broth that stayed sterile forever. And so it turned out Louis Pasteur really needed help to make sure that this point was driven home. Patrick, do you have ideas on what might have happened there?
After he evacuated it, all that positive pressure inside, did he keep it at room temperature?
Yep.
I was thinking if you cooled that, you would create a negative pressure in there and it would draw the air back in.
Yeah, well, he really just set it onto the bench top.
Yeah, so
yeah.
About like other scientists might not have been so careful with maintaining the pressure.
Right, right. Good. I love that. So what if, you know, you had some kind of a pressure where it was actually pulling in the microbial life that was in that first U-bend, and this was causing, I love that. That's wonderful thinking about like what could have gone wrong when it was tried to be, when scientists tried to repeat it. Can anybody think of another thing that they could have run into? Other scientists trying to repeat his work. Where might he have fallen down here?
They probably didn't sterilize the broth.
Good, good. So what if you didn't use a rigorous enough procedure to sterilize the broth? Like, could that have been the fall down in all of this? That's a great point. And in fact, as it turns out, other scientists didn't even use the same type of broth that he was using. Yeah. So if you think about this, if you had a broth that you took from a cow, right, beef broth, and then you compared that to, say, broth that you made from hay, like we call that a hay infusion, what's going to be very different in those two broths? Yeah. So the microbiome, if you will, of a beef extract is going to be very different than the microbiome of a hay extract. And in fact, in hay, we find these crazy, awesome structures called endospores. Endospores are these wicked, dormant, resistant structures that can live through almost anything. And unless you sterilize the broth properly, you're not going to kill them. And so this is why he couldn't repeat that, why other scientists couldn't repeat that experiment. Awesome. You guys are doing science. So that's fantastic. So let's just write down briefly what needed to happen in order to really put the nail in the coffin of spontaneous generation. Pasteur's work needed to be repeated. And you remember that passage I read to you from the work of John Tyndall? He came on the scene with this, and he helped a little bit by saying, hey, it looks like different people are using different broth types. What might need to be true about how we sterilize different broth types? Do we need different boiling times? And so, yeah, some microorganisms are killed with a short boiling time, whereas others are heat resistant. And in fact, one of those heat resistant structures are endospores. So it was in 1876 that finally the nail was firmly hammered into the coffin of spontaneous generation. And everyone started to believe that life comes from other life. So, speaking with your neighbor, who's willing to share what their answer was for that question? Correct? What'd you say? See, about 140 years ago, which is nuts, right? Did anybody think it would have been longer ago? that we, yeah, one would think that it wouldn't have taken that long to realize that life comes from other life, but it really wasn't all that long ago, at the end of the 19th century, that we really saw that lay to rest spontaneous generation. What's really interesting is that year, a little bit later that year, a man named Robert Koch began to do some really cool research where he was looking at one of the most famous endospore-forming bacteria called Bacillus anthracis. So Bacillus anthracis, I heard what does it cause? Anthrax, you got it. And I know I feel older and older every time I mention this, but you guys remember back to 2011 or 2001, 9/11, when we had the bioterror scares? Who remembers that? Yeah. What was happening with the whole bioterrorism thing? You remember, Riley? Yeah. Yeah, yeah. Right, any life. But of course, endospores are also pretty, resistant to a lot of that. So that's exactly what was happening. And just to reiterate Riley's words over here, in post 9/11, there was a bioterrorism scare in which there were envelopes being mailed with highly purified endospores in them. It looked like a white powder because the endospores were so pure, but really it was resistant dormant microbial life inside of those envelopes. So when people opened up the envelopes, they just breathed in a massive amount of endospores. And of course, those lodged within the lungs of the recipient. And when they were in the lungs of the recipient, what happened? Yeah, go ahead, Brad. Yeah, they grew. And I hear you saying too, it's really cool. Endospores actually go through this process of activation when they hit a new temperature, and they get activated in almost like a popcorn kernel. They almost pop open. And then they begin to grow, and they get what we call outgrowth at that point. And then they cause pulmonary anthrax. So a number of people actually got, and some died from, pulmonary anthrax. So that was one of the most interesting things in our memory related to the early work, which it's crazy to think that it was all the way back in 1876 that Robert Koch actually determined that Bacillus anthracis causes anthrax. So here's what we're going to do right now. I want you to write down the 4 things that you would need to do if you were going to show that a bacterium causes a disease. For example, with Koch's early work, he was showing that Bacillus anthracis causes anthrax. But it could be anything. If you wanted to show that a bacterium causes a disease, what would you need to do? And there are 4 of these things. So work with that neighbor and write down the 4 things you think you would need to do, and be noisy as you'd like. That's a great question. The question is, are these ethical things? That is debatable. Have someone who writes legibly actually jot these down. That would be super awesome.
Yeah, like cake.
Good.
And that will only be in the environment, because if there's something else.
Good, good, good. Good. I see a few groups that have hit 2 things. Has anybody gotten to 4? Okay, Hunter's like, he's raging. Okay. Try hard. Sweet. Cool. Do you mind if I share what you've got there?
Nope, go for it.
Sweet.
You can just, and it picks up like that. So.
Sweet. Got it. Got it. Got it. Got it. Yeah. Thanks. Perfect. Okay. Well, I'm hoping our doc cam will come up here. Don't give me that. Okay, well that's all right. I'm just gonna, oh, there it is. Oh, perfect, yay. Huh. So this is Hunter's beautiful work. Let's see if we kind of all came to some of the same conclusions. So number 1, he says we need to isolate the bacterium. Hunter, what does that mean? Yeah, and so I love that. I couldn't have said that any better. This semester in lab, we're going to be doing a lot of making sure we only have that one thing. And that is what isolation means. This is great. This is one of Koch's postulates. But what would you also need to add to that to give it a little bit of context? Isolate it from where? From Jake.
Could you just like basically this would like contain his thing? They set up an experiment with control.
Okay, good. Always a good idea to have a control. So in this case, right, we want to know if the bacterium causes the disease. So where would we expect to find the bacterium? Yeah, in the infected host. So before we would isolate, we would want to say that we had an infected host, right? That we had a diseased organism. So from a diseased organism, we would want to be able to isolate the bacterium, right? So that is perfect. Now you're going to have some code in here that I can't get into, aren't you? Okay. 2, 5, 8, 3. Yay, got it. So isolate the bacterium from the diseased host, okay? So this is important, and we would want to make sure that the organism, we would want to make sure that the bacterium was only present where? In the infected, okay, so now here's where Jake's control comes in, right? The control is a healthy host. So we would want to be able to isolate it from a diseased host, but not from a healthy host, right? So present in the diseased host, but not in the healthy host, and we isolate it from the diseased host. Okay, then I love this. Number 2, he has administer it and it causes disease. So let's add just a little bit of detail to that one as well. So administer it to what? The healthy host. And see if the disease ensues. Yes. And this is where Riley's questions of ethics come in, doesn't it? Yeah, hit me.
So what if the healthy organism had something that could like fight off?
Right. So there are some really interesting caveats to Koch's postulates. Because what if the healthy host is for some reason resistant to that particular disease-causing pathogen. So there could be situations in which that was true. Does anyone else have a critique of something we've said so far as well, based on your reading about the microbiome? What might be true about the caveat that we said that it's healthy, or that it's found in healthy hosts, but it's not found in a diseased host? What's the caveat, Patrick? Okay, yeah, kind of, but maybe it is just present. Is that what you're thinking? Good, 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 right. So we also now know that in many cases, we have pathogenic bacteria in and on us, but in the right balance, they don't cause the disease. Sometimes we call that a carrier, or sometimes we just say that it's part of your microbiome, but unless it gets out of whack, it doesn't cause disease. And so there's a lot of nuances that we now understand a little better about Koch's postulates. But yeah, no, this is absolutely fantastic, and I totally didn't memorize that at all, Hunter. Okay, you see you've got the, don't steal his iPad. See, my problem is I start typing in my code. Cool. So good. So then I like this. I like this idea of like being able to somehow remove it and cause a lack of disease. But what's the problem with that sometimes? Yeah, it's almost impossible to do. One thing that we would put next is actually that after we had re-administered it to a healthy host, we would actually want to re-isolate it from the healthy host that became diseased. So in microbiology, we always isolate and re-isolate. So that's something to keep in mind. So if we were to kind of knock this out, what we would say is that we would start by seeing whether, noting if the microorganism was present in a diseased host but not a healthy host, 1, right? 2, we would isolate, okay? 3, we would reinfect into a healthy host and see disease symptoms. 4, we would re-isolate, okay? So I think you did an awesome job. Let's see if Patrick came up with some of the same postulates. Yeah, isolate, okay, and then inject the dead bacteria. See, this is like a control, right? Infecting trying to see that there isn't a disease if the bacterium is dead, whereas if it is live, it causes disease, and kind of like that control. Okay, cool. Does that make sense? Do the nuances that we now question in those postulates make sense? Okay, sweet. Yeah, so number 1 is it present in an organism with a disease and absent in an organism that doesn't have a disease? Yeah. Yeah. Yeah, of course. Thank you very much for your awesome participation in that. Okay, cool. So we're going to switch back over here. And okay, so 1, what's 1? Present in a host that has the disease, right? But absent in a healthy host. Okay, number 2, what's that? Isolate it from the disease host. Okay, good. What's number 3? Cause the disease in a healthy host with that isolate. Okay, and then 4, re-isolate. Good. Yeah, thank you. It's always just a confirmational step. Yeah. 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 Yep. And we do a lot of that, confirming, you know, it's a lot of just making sure, like when we're looking for, say, a pathogen in water, we'll confirm and confirm again and then confirm a third time just to make certain. Wonderful. So last night I was enjoying reading through the posts on the Follow Your Gut book discussion, and Jade asked a wonderful question. How and in what ways do microbes play essential roles in humans' lives? What would happen to people if microbes suddenly didn't exist? And this question is beautiful, Jade. I don't actually think did I ever end up meeting you? Not really. Okay, wonderful. A beautiful question, and perfectly segues into the next thing that I want to briefly touch on, and that is the applications and implications of the microbial world. And I thought that it was possible that as we begin this, Sarah might talk a little bit to us about your passion about the ocean. What is one of the things, like with regards to element and nutrient cycling, the roles that the ocean plays?
Well, one example is that it absorbs a third of the CO2 in the atmosphere.
Perfect. It's like an epic carbon sink, the ocean. Yeah, sweet. And? Sorry, I don't want to cut you off.
No, that's fine. It also breaks down a lot of things that we put into it.
Yeah.
To the organisms that take it in at the surface level,
Mm-hmm.
all the way down to
Yeah, like a gradated nutrient cycling process. The ocean is epically good at contributing not only to carbon fixation, but also oxygen fixation, so the generation of oxygen through organisms that actually live in a photoautotrophic way. What else, Riley?
As sediment from the deep ocean are washed into shallower, warmer waters,
Right.
algal blooms happen, and
Mm-hmm.
producing a great majority of the primary.
Yeah. Good, perfect. So so when I say something is photoautotrophic, that's another way of saying that it's a primary producer. So it's a perfect way to say again that role in not only as a carbon sink, right, sucking up CO2, but also generating O2. I'm playing a magnificent role in that. Obviously now, I love that you bring up algal blooms, because if the balance of that cycle is disrupted, and what's one of the things that can disrupt the natural cycle of algal blooms and then lack of algal blooms? Yes, oh my God, you guys are amazing. Yes, fertilizer runoff. We talk about what we call NPK, which is nitrogen, phosphorus, and potassium. These are things coming not only from fertilizers, but our phosphates coming from detergents and things like that. So we are seeing that sometimes we get massive algal blooms, which also disrupts that cycle. But I love that. So broadly, I want to state this, that microorganisms are key, right? Not just algae, but also bacterial life and other forms of microbial life in converting things like atmospheric nitrogen into forms of nitrogen that can be used by plants. This is termed the nitrogen cycle, one that we will talk about more later. They replenish atmospheric oxygen. They decompose materials that no other organism can degrade. And holistically, they are at the base of food chains and nutrient biogeochemical cycles. And you all have given some beautiful examples of what that tangentially means, or tangibly means. So huge. And I guess, Jade, if we were to give like that really broad answer to your question, if there was no microbial life, everything would die and nothing would even decompose. And I think it's weird to think about a situation in which that would happen. Yeah, yeah.
So this is kind of going back to what we talked about earlier.
Yeah.
And we're just moving.
Yeah.
So I want to ask before.
Sure, please.
But I'm just finding it really ironic. I'm a biology major.
Yeah.
That we are laughing at the idea of spontaneous generation now, but everything that I've ever been taught in all of my biology classes about what almost, I would say probably at least half of the scientists around the world hold to, as far as evolution goes, is the exact same thing.
Yeah.
People still believe that life can come without.
Yeah, absolutely. Absolutely. Yeah, no, and there are a lot, and that is one of the reasons that I make that point, that we are still polarized, right? And we are still polarized on super detrimental issues. And I would love, this particular class seems to me like a group that might also want to pick up another book that I've asked for as a optional reading called Fevered, and it's written by Linda Marsa. And to me, this is an issue where somehow we manage to maintain polarity in the issue of climate change. And it's crippling us with regards to changing anything because we have that polarity. So I love that you bring that up on a really meaningful level. Yeah.
So I'm just kind of wondering, I don't know, maybe physicists would differ on this, but the formation of the solar system, you know, and life really did arise out of, you know, inorganic and basic organic.
Yeah, okay, good. I'm so glad this question came up because so we sit here and we talk about like putting the nail in the coffin of spontaneous generation, not a thing, right? But life had to arise. So this is actually an important question because what was the first abiogenic event? And right now, one of the most favored hypotheses is one by a man named Walter Gilbert, and it's actually a hypothesis called the RNA world hypothesis. Has anybody heard of that? Yeah, so this is actually a pretty cool hypothesis, which states that RNA, because as we've recently discovered, RNAs can act as enzymes. They actually can be catalytic. So that is crazy. And when you think about the fact that an RNA actually also can hold and store the genetic information, an RNA, in theory, could be both the worker, the catalytic macromolecule of the origins of life, and also, at the same time, be storing the genetic material. So it might be that DNA then evolved later from that catalytic RNA, and that the catalytic RNA was kind of at the root of the origins of life. And so that's one of the primary and preferred origins of life hypothesis right now. So yeah, sure, there had to have been an abiogenic event, didn't there, at some point. We just know that now it doesn't happen all the time. Like there was something crazy that went on that allowed that, yeah. Yeah.
How did it, in our ideas?
Right. But what's really cool is there's been some really cool benchtop research where they've actually been able to create from inorganics alone proteins. Yeah, polypeptide chains, right, which is super, super cool. So next time, here, let me tell you before I can tell you're getting restless. So next time we have some really cool things going on. Number one, I'll finish off what we're talking about right now. But number two, you're going to hear about the gaming group option. That may be something that appeals to you. Number three, we're going to have a guest lecturer on the human microbiome. His name is Christopher, and he's totes rad. So come on Friday to get all that. Fevered, and Linda Marsa is her name. And I might even have an extra copy if you grab me soon and that I could lend you.